Well, good afternoon, everyone, and welcome to the first talk of 2018 for the Network Speaker Series. My name is Jan Amen. In case you don't know me, I'm the director of C-DEBI. And for those who don't know, the Network Speaker Series is a C-DEBI thing designed for early career investigators, and the speakers are nominated by members of the community for the exciting research and also for their effective communication skills. And before I officially introduce Alma today, let me remind people that we have 3 more talks lined up for this fall. On September 20th, we'll hear from Nagisa Mahmoudi from McGill University. She will have just moved or will about to be moving to McGill. Just 1 week later, on September 27th, we'll hear from Jackie Gorial from the Bigelow Lab for Ocean Sciences. And then October 18th, Rosa Leon Zayas from Willamette University. All the talks are on Thursdays at exactly this time. All right, so Dr. Parada received her BS in biology from the University of La Verne. She then attended the University of Southern California for her PhD, where she worked with Dr. Jed Furman, studying the temporal and spatial variability of marine archaea at the San Pedro Ocean Time Series Station, about 10 kilometers or so off Los Angeles. She is currently a C-DEBI postdoctoral fellow at Stanford University, working in Dr. Ann Dekes' lab, where her work focuses on studying the diversity, distribution, and role of benthic archaea in the carbon cycle using combination of molecular and stable isotope probing techniques. Her talk today is entitled Evaluating the Diversity and Distribution of Novel Microbes Across Physical and Geochemical Gradients in Deep Sea Sediments. And before I turn the microphone over to Alma, let me just remind everyone that you can participate in the Q&A by typing in questions into the chat box. And then after the presentation, which should be about half an hour or so, Alma will answer as many questions as she can or feels like. So with that, I'll turn the microphone over to you, Alma. Thanks.
All righty. All right, well, thank you. And thank you for watching and joining in. So like Jan mentioned, we're talking about microbes and looking at the diversity and distribution across different geochemical gradients. We're going to be talking about mostly the entire community today, not just archaea, but we will be having some kind of cool things coming up about the archaea a little bit later on. And most of this will be work on the molecular side. So let's go ahead and get started. So first, marine sediments. What is that? Like, what are we talking about? We're talking about that. And what we're talking about is the area, essentially the seafloor. This is encompassing from the sort of shallow water depths on the continental shelf down into the abyssal zone, including not only the very superficial part of the sediment, but also down into the deep subsurface. So because we're talking about such a large area, these marine sediments cover over 75% of Earth's surface. Meaning there's a lot of places for microbes to live. So these sediments, including the deep subsurface, hold about 2.9 times 10 to the 29th microbial cells. This is about 50% of all microbes on Earth, actually. So if you were to look at sort of what type of environment these microbes are experiencing, what we're seeing is that with distance from shore, not only are they experiencing higher organic matter in the shallow sediments and going losing that organic matter as we go away from the shore and deeper into the water column. We're also seeing decreases in temperature, but at the same time increases in pressure and the availability of oxygen into that sediment location. So what does that mean? Is that these microbes are essentially, really, if we would take a look at the core of the sediment, so like a cross section, they may be getting oxygen, but sort of really only at the deepest part of the sediments themselves or into the water column. And as that oxygen is being depleted, we're selecting for different types of microbes. So these are microbes that are now at the shallower part capable of aerobic respiration. But as this sort of oxygen is being depleted, we're selecting for different types of microbes, having different metabolisms, such as denitrification, sulfate reduction, as well as carbon dioxide reduction. So again, the environment is selecting for very different types of microbes. So it kind of poses questions as to where are these microbes living and have we seen them before? So one thing to remember is the majority of the benthic space lies really deep, with 98% of the ocean volume really lying below 22,000 meters in water depth, and the average depth of the ocean being 4,000 meters. So really we need very different types of sort of strategies to be able to look at it, from Niskin bottles to get really down deep in the ocean if you're interested in the water column, also multi-cores and submersibles. This really large area is really important because even if we're looking here on the x-axis, the water depth, and we're looking at sediments in very shallow depths, as we go deeper into the water column, the top 1 centimeter sediment is still holding the same amount or abundance of microbes in that top centimeter. So even though we're pretty deep into the water column, we're still having large microbial presence there. However, estimates suggest that only 5% of these deep oceans have even been explored, with less than 1%, much less than 1% ever been sampled. So really opening up a question about, well, if we've undersampled this area so much, have we ever seen a lot of those microbes? How well characterized are microbes that are living there? And that's really sort of a basis for the research question and motivation, is looking at what is the variability of these, then the diversity and distribution and activity, and in the end, what are the metabolic capabilities of microbes living across these different physicochemical gradients spanning representative depths of the ocean. And this really can lead us to understanding sort of what are these microbes that are there? Have they been characterized before? And what is their contribution to global biogeochemical cycles? So to that, we looking at the microbial communities on the transect off the coast of San Francisco. This is San Francisco here, and sort of the beginning of a transect. So their transect not only covered areas that were close to shore as well offshore, but if we look at the cross-sectional view of these of this transect, we see that we sampled areas that were 100 meters down to 4,500 meter water depth. So again, kind of not only getting into how did the microbial community change with distance from shore, but also with depth in the water column. These are multi-core samples for the most part. So these are cores that are about 30 centimeters in length. We also took gravity cores at 3 sites along this transect, which I'll discuss a little bit later. So these are cores that can go up to 2 meters in length. So to look at the microbial community, we were able to subsample from these cores different horizons. So that's what's depicted in this image here, to be able to get at looking at the microbes at different horizons across this core. We also took subsamples for pore water to be able to look at the chemistry that these microbes may be experiencing. So to look at the microbial community, we took subsamples between each of the horizons were either 2.5 or 5 centimeters thick, and extracted the DNA and RNA, and looked at the DNA essentially to look at the presence of the different microbes and examine the activity of the RNA, activity of these microbes using the RNA that was extracted. Then we are able then to amplify this DNA and the RNA using universal primers to really be able to get at the bacteria and archaea in these samples simultaneously. Then sequence these using aluminum iSeq 2x250 base pair technology to look at the sequence of these 16S RNA, and use bioinformatic tools to cluster these different sequences together to be able to obtain operational taxonomic units. As we all know, species isn't really something we want to say with bacteria. We don't really know how to delineate that, but we can look at sort of the most base level, so individual OTUs, and classify those on taxonomy to be able to have an idea and compare like what are the different phyla, class, and so on found in different samples. From that, we can create an OTU table, so we can look at individual OTUs and how their percent relative abundance changes in different samples. So when we apply that to our transect, and we look at the microbial communities at each of the different water depths shown by these different panels here, and look at the percent relative abundance of the 10 most abundant phyla across these water depths and across the sediment depths on the y-axis. What we see is that if we just sort of focus on the colored bars without trying to match the phyla yet, we see that sort of the beginning of the transect from 100 to 2,000 meters, the communities don't really change that much with sediment depth. We see some sort of small variation, but it's not huge. If we move into the 2,500 meter and further into the water depth, we see that these communities really do begin to change, and we start seeing very different phyla becoming more abundant as we go deeper into the sediment. With the largest sort of differences overall in the communities happening at the deepest water depths, where we see not only a shift in the overall community due to water depth, but also within the sediment itself. If we look at the colored bars, you would see that the majority of our phyla or abundant phyla are actually bacteria. But with the archaea, we do see, it's kind of harder to see here within sediment depth in the shallower water depths, we do see increases in the percent relative abundance of archaea within sediment depths. But we really do begin to see these archaea becoming more abundant deeper into the water column. So we are able to see that there are changes happening within the community across this transect, not only with water depth, but also with sediment depth. We wanted to see, well, what it could be potentially contributing to these changes that we see, or what environmental parameters are correlated to these changes that we're seeing in the microbial community structure. So to do that, we can do what's called a CCA, or canonical correspondence analysis. This is an ordination plot that allows. So in this case, we have individual samples represented by the circles, and the color of each circle is given gives a different water depth. And the proximity, or how closely clustered individual samples are to each other, tells you how similar they are. So if they're really close, they're really similar. The further they are in this ordination plot, the more dissimilar these communities are from each other. And the vectors here are giving the correlations and the different environmental parameters to how these communities are distributed in this ordination plot. So if we first take a look at water depth, we can see right away that the communities appear to be clustering based on what water depth they come from, with sort of the shallower samples, or water depth samples, appearing here on the left. And as we move sort of towards the right into the ordination plot, we start beginning to see the water depth, showing that there is separation in these communities based on water depth. If I had labeled these according to the horizon or the sediment depth these samples came from, you would actually see that these samples down here come from shallower sediments, with the deeper sediments clustering, or the samples from the deeper in the sediment clustering here towards the top along the y-axis. If we look now and relate that to the environmental parameters that we measured, we see that temperature, pressure, and the oxygen concentration of the overlying water, so the water that's lying on top of the sediment, seem to be contributing or are most correlated to the distribution of samples across water depth. While concentrations in pore water of ammonium, bromide, nitrate, and the oxygen content or concentration of the pore water being most correlated to how communities are distributed within the sediment itself, suggesting that sort of these environmental parameters are correlated to how we see these communities changing across the transect and across the sediment depth itself. So now that we saw like these communities are different, we are seeing sort of they are responding or are contributing to how their environment is selecting for each of them. We wanted to know, well, what are the organisms right here? Have we seen them before? Like, are these organisms been characterized before? Like, are we now finding, all right, we're selecting for organisms that are really different, as seen here by the 4,500 meter being quite different from the rest of the samples. Like, are the organisms here the same as those we find there? How similar are they, and have we characterized them before? So to do that, we compared the representative OTU sequence for each of those samples to the 16F of reference genomes in the Genomics database. And what we did was we actually looked at the percent identity between the representative sequence and the 16S reference, or the 16S from the reference genome. And on this plot here, we have along the x-axis the normalized percent identity. So this is a percentage, essentially the average percent identity for all of the OTUs in a given sample, accounting for the abundance of each of those OTUs. And on the y-axis, we have sediment depth. And what we see here is first, I should point out that I have also indicated sort of where you might see differences in, different genus or different family going to the left, or different orders or phyla. And what we see is that the shallower depths, down to about 20 or 15 to 20 centimeters depth, really are representing new genus. The average OTU in these samples belong to a different genus from those of the reference database. While as we go deeper into the water column, we start seeing that the communities that are in these samples likely are quite different from any of those that we've sampled before. So they are quite different from each other, with sort of the deepest samples being even more uncharacterized. So potentially, at least at the 16S level, these are different from what we've seen before. So signifying the potential for novel genomes. And I'd like to point out that I did include the 3 gravity core series that I discussed before. So that was given by the stars. So if I include the deepest part of the gravity cores, so again, this is the area that was shown before with the multi-core depth, so 0 to 30 centimeters. We see that the majority of the gravity cores are actually all OTUs that are potentially belong to very different families from those that we see in the reference data sets, showing again there's a really high potential for novel genomes and in this transect itself. And one sample that I'd like to sort of really point out is this one here. This one is the sample from the gravity core at 1,500 meters at the 25 to 30 centimeter sediment horizon. That has an average, essentially, percent ID that puts it potentially a different phyla, or at least a different order. And this most likely, well, these kind of brought to our attention, wanted to know, is there a specific taxa, or is there a group of OTUs that might be skewing this average percent identity that's pushing it so far this way, since it is essentially an average scale to abundance. So to do that, we look really further into this sample and trying to see, well, what organisms here could be skewing this percent ID. And one really interesting thing is that we actually had to turn to the archaea. And this is once again, a bar plot showing you the percent relative abundance of the 50 most abundant archaeal OTUs across the transect at 1,000, 1,500, and 3,000 meters. And again, on the y-axis, we have sediment depth. And what we can see is that the archaeal communities do change between these water depths. And one thing that should point out to you, or come out to you right away, is this giant purple bar here that actually belongs to the phyla Wozoarchaeota. And what if we're able to show you a little bit further here, this big bar here is actually dominated by a single Wozoarchaeota OTU. And this OTU, when we compare it to other reference, or actually, I guess, other complete genomes that we have of Wozoarchaeota, none of which come from deep sea marine sediments, they really aren't similar at all. They're very different from each other, which is really causing that skew in average percent identity. So one of the things that we are planning on doing in the future is to do some metagenomics, and Yuna, a master's student in our group, will be performing or trying to get metagenomes from this to be able to hopefully characterize the Wozoarchaeota that we have here and look at what could be happening at this sample to be, or this site here to be selecting for such a high abundance of Wozoarchaeota. So it'll be really interesting to find out kind of what's going on there and what type of Wozoarchaeota are we finding. Oops. So that kind of leads into the idea of connecting identity with function. We're able to see that there are Wozoarchaeota there, but with 16S, all we really see is identification. We have lots of reference databases that allow us to tie these 16S sequences to an identity, or essentially a classification. But as most of us probably know, that the taxonomy is really just not a very good indicator of metabolic potential. There's lots of other genes that are in a genome that really makes an organism behave quite differently, and from other things that may be classified similarly. So we have to turn to metagenomic sequencing. This is a sequencing of all the genes of all organisms in a community. We can take all of those sequences and all those genes and assemble them into what's called metagenome assembled genomes, or MAGs. And if we're able to obtain the 16S ribosomal RNA gene with that MAG, we can then connect identity with function, right? Now we have the power of all these reference databases, so we can match our 6DS to what we've seen before and get an idea of the taxonomy of a particular mag, and then look at the rest of the genome to get an idea of that metabolic potential. Like, what are the genes? What could they be capable of doing? But there's a big problem with that. One of the biggest challenges is that typical methods really are not able to obtain 16S genes consistently with these mags. And the main reason for that, these are highly conserved regions. And it really makes sort of current assembly methods really difficult to get these 16S RNA genes, or at least not very many of them. So one solution that we have sort of dove into is using the 10x Genomics and Athena technology together in collaboration with Ami Fox Group at the medical school here in Stanford. And this takes advantage of, first, the 10x Genomics. And this is where we get a metagenome, we select for high molecular weight DNA, use microfluidics to partition these long fragments into individual droplets, then create essentially short reads based on each of these long fragments that are barcoded. And that's really the important part, because that means that we can now match these short reads with these barcodes back to what fragments they came from. And the reason that's important is that we can then use all that information using the newly developed sort of Athena assembler, which first takes advantage of common assembly methods, which like seed methods that use de Bruijn graphs to build a contig or continuous sequence. So essentially, this is cutting these short reads into shorter Kamer sequences that are pieced together based on their overlapping sequence to create these contigs. So these create these really awesome looking scaffold graphs. And what's happening is that you're building your sequence based on your short reads, depicted in this sort of top panel here. Problem is that once you get into these conserved regions, right, these are the regions that a lot of organisms have very similar sequences here, it's really difficult to decide sort of how, if this sequence belongs to sort of this part here or not, because you really can't resolve how you should extend this contig. But now we can use the barcodes on these short reads to map it back to these longer fragments that are in the microfluidic partitions to be able to decide, all right, these sequences go together, so clearly this sequence should have been built this way. So then we can take the shorter contigs that created from the seed assemblies, along with the longer scaffolds built from using the sort of the novel part of the Athena assembler, put these all together and reassemble everything. And this really helps us sort of resolve breaks in these sequences, allowing us to build longer contigs and longer sequences. I should say from this, we were able to apply this to one metagenome from 3,500 meter and the 0 to 45 centimeter horizon, and kind of really were able to build a lot of 16S sequences. We had 64, I think it was 64 total 16S sequences obtained through Athena, versus I think it was like a fraction of that. It was 16S sequences total from using seed assembly alone. And much longer, an order of magnitude longer contigs were obtained with Athena versus the regular seed assembly using megahit. So with that, then we were able to look at the mags and look at more of the nearly complete mags. So if we were able to obtain 24 mags that had over 70% completion and a 16S ribosomal RNA gene from this one sample. And most of these actually had 98% similarity to OTUs in this transect. So we essentially matched the 16S from these mags to these OTUs. And 22 of these mags we found were potentially widespread. So unfortunately, this all came up to you together. So hopefully this doesn't scare you that there's this huge graph, but I'll walk you through this top panel. This top row of bar graphs are showing you the percent relative 16S ribosomal gene abundance in constant transect. So these are looking at the percent abundance of the OTUs that matched these mags. And what we're seeing is that these OTUs are quite widespread, with like a lot of the sort of the beginning of the transect selecting for OTUs matching mags from the sender-SENACE group. and some shifts happening at the sort of deeper water column depth, suggesting that there are at least OTUs that are very similar to the 16S of these mags across the transect, despite this metagenome coming from a single sample in the surface of the 3,500 meters. If we look at the 16S ribosome RNA abundance, we see a slightly different story in which we still have the Sediments being most abundant at the beginning of the transect, but we have different taxa being selected for in the RNA, suggesting there are different activities of these OTUs across this transect. One that kind of points out to everyone and jumps out is this large blue bar, which belongs to OTU from the Nitrosamonidaceae. So being able to connect that 16S of the MAG to the genes, we see that this MAG or this OTU is potentially involved in denitrification. The pink bars here are representing the set of another MAG that is, or OTU, I should say, that is in the top 3 in abundance across this transect, belongs to the disulfide. And looking at its genome, we see that this is involved in sulfate reduction. While the Sediments seems to have mostly genes involved in sort of general heterotrophy, suggesting again that these different environments these OTUs or these organisms are being exposed to is selecting for different metabolisms, especially what is active there. So eventually we'll try to go back to the RNA and build, potentially look at transcriptomics to see, do we see sort of this mirrored in the transcripts for all the genes? Like, are we selecting for different genes involved in different metabolic functions across this different transect? But really being able to match the 16S of these MAGs to these OTUs really lets us take advantage of all the sequence information we get from amplicon data back to the MAGs. And again, the more 16Ss we can get binned with our MAGs, the more we're able to do this. So we wanted to look at sort of the more complete MAGs, so the things that are nearly draft genomes. And in this case, we looked at we were able to obtain 9 MAGs that had greater than 90% completion and less than 5% contamination using check-M. So this is looking at the percentage of single copy genes that are in a given bin. And we were able to, like I said, get 9 bins that were nearly complete. And actually, we had 0 MAGs that had that level of completion when we did seed assembly alone. And because we were able to get the 16S with each of these bins, we were able to look at the Silva taxonomy based on 16S, once again, taking advantage of those reference data sets. And as well as looking at how does that match with the 16S, how does it compare to the 16S of representative or reference genomes in the GenBank database. And so we were able to look at the 16S and compare that and look at the percent identity. And what we see is that we get any range where from 80% to at most slightly under 95% identity, kind of hinting at that we are getting, or we have sort of seen organisms with similar 16S sequence before, but they're still quite different from each other. So sort of look at, does that reflected in the genome itself, or are we seeing genomes that are more different from each other? So for that, we looked at the average amino acid identity between the bin and the representative genome. And this is looking at a minimum of 50 genes between each of these genomes or bin. And we found, again, that mostly we're in the 40s of percent average amino acid identity, suggesting that we are looking at quite different genomes between each other, even if they are potentially related. We're probably somewhere near the family or order level difference from each other. So these new bins are potentially new family members, essentially. So if we look now at the potential energy source, I realize that this data table here is actually an older version, so please ignore some of this and just focus on what I say. So the potential energy source for some of these are mostly anaerobic respiration, with some of these groups, like the gamma proteobacteria here, potentially involved in denitrification, and another one in sulfate reduction. But all of these potential carbon source for these bins appear to be organic carbon, which is a little bit different than you might expect, because like I said before, this is a 3,500 meter site where we're expecting to see lower amounts of carbon compared to sort of earlier in the transect. So we were really interested, why would they be getting organic carbon? Like, why is that your like preferred carbon source? So to do that, we looked at the total organic carbon in our samples, and we're able to do that for a subset of our samples. So on the x-axis, we have sediment depth. On the y-axis, we have organic content. And what we see is that the 2,000 meter and 3,500 meter sites actually have the highest amount of carbon, or organic carbon, in these samples. Most of the transect lies far below that. And of course, the sample from which we obtain a metagenome from has the highest amount of organic carbon. So suggesting that, they have some sort of input of organic carbon here that we weren't really expecting. And that may be selecting for these organotrophs as indicated by our mag. So really future research, or at least one of the focuses, will be looking at, well, where is this carbon coming from? How much carbon is really here, especially throughout all the rest of the sites that we have across this transect? And what is the composition of this carbon? And relating that back to the distribution of organisms that we see, like is there a reason why we're seeing these shifts that might be related to carbon, as opposed to some of the other chemicals that we measured in the pore water? So with that, I kind of to conclude and some future directions. First, we did see sort of like a lot of variability in the distribution of taxa across this transect, suggesting distinct adaptations to those environmental conditions. So one of the future directions really be looking at the genes that may be contributing to the selection of this taxa, especially within metabolic guilds. If they are, for example, if you have two organisms that are using the same electron acceptor, but we see one at the beginning of the transect and another one sort of later on at the deeper water depths, sort of what are the differences between these genes that may be contributed to this selection? Or looking at some of the other genes in their genomes, if there's like differences in pressure, like do these organisms have genes that might be helping them stabilize like their membranes, for example. Additionally, we are seeing sort of a widespread distribution of potentially novel organisms, suggesting that these areas really are undersampled. There are lots of previously uncharacterized taxa here, and it's really sort of important to further characterize these genomes to determine their metabolic capabilities, so that we can get a better idea of sort of what is the potential for contributions to the global biogeochemical cycles. So it'll be a lot of fun to haven't we seen before. So with that, I'd just like to acknowledge my PI and mentor, Ann Dekas, as well as co-authors, the rest of the Dekas Lab, and funding from NSF, C-DEBI, and Stanford University as well. Thank you. And I will wait while some people have time to type any questions, and then I'll go scroll through those and answer them as I can. Well, I see the ORCID Lab asked a question that I will go ahead and try to answer while the people are going. So I would like to say that I accidentally uploaded the wrong PowerPoint. So I'm sorry if you got really excited about the methanogenesis. We did see some genes in those bins, so that's why it was there to begin with. But it wasn't complete pathways. It seemed to be, I can't remember right now the region. So the question from ORCID Lab was, do you see evidence for a complete methanogenic pathway in your data bacteria genome bin? And like I said, we see, I believe it was like 2 or 3 genes that were involved with methanogenesis in those bins, but really I want to kind of go back and sort of curate that and make sure that that belongs there, because that would be kind of a crazy, amazing thing to see, having these bacteria have methanogenic capabilities. So I really we'll need to go back through that and sort of manually curate those bins. And then if a really awesome paper later comes out, then you'll know kind of what I felt about that genome bin and that capability. The next question I have is from Jan, is that CCA analysis showed that the deepest 4,500 meter sample is by far the most different, even from 4,000 meters. And why? So one of the reasons, well, I guess there's a number of reasons. If we look sort of at the profile, we're actually much further away from shore. And maybe if I can eventually scroll back over there, but maybe I won't try too hard right now. We are much further offshore. So again, this transect is not only covering differences in water depth, but we're also distance from shore. So kind of looking at sort of what environmental inputs to this area might be sort of changing and explaining why you might see differences between just 500 meters in depth. So I suspect, and one thing I'm not showing my CCA is sort of the variance that's explained by these variables. And it's actually quite low. For the variables themselves, they're only about 30%. So I think that we really are missing some things that might explain why we're seeing such differences even between these two samples. The 4,500 meters is also where we're seeing the most archaea. So that might be a really interesting thing. Like there might be something there that's selecting for archaea, and that is contributing quite a bit to that change that we see in the CCA between those two depths. Scrolling back up. I don't know how to say this person's name, but I got a question of when you match the OTUs 16S and MAG 16S, do you map the reads or do you match by matching taxonomy? I actually did a blast between the representative sequence and the 16S. So I just did a blast between the two sequences, and that's how I matched them. So again, that's the awesome thing about actually having a sequence. I could actually compare the sequences of the two things together to be able to match that up and look at the taxonomy that way. I have another question. Did you confirm that these 16S and MAGs are not contamination and validate with methods to confirm the taxonomy assignment? I looked at some of the random bends and looked at some of the ribosomal genes for some of them, and looked at other ways. So like Parada also gives you like a taxonomy assignment based on other genes, not just the 16S. And they pretty much match almost every time. There's never anything that popped out that said like this taxonomy doesn't match at all. So I think that it is quite real. I mean, we'll keep looking at it, and as we kind of develop this a little bit more and build phylogenetic trees really based on not just the 16S, but to really confirm that that's true. But so far, it seems that it really is working, that those 16S really do belong to those MAGs. So it's a really cool thing about this sort of way. Next question was, I was wondering whether there are any visual features of the sediment for the high carbon deep samples that might illuminate the question of carbon source composition. Did you do any further physical characterization of the sediment? The answer is not yet. We are going to go through that. We're currently trying to go back to the lab notebooks because we do remember seeing differences in color of the sediment. We do have sediment samples still stored that we're trying to go back to, not only to characterize the carbon, but maybe porosity has something to do with that. If there are any other sort of chemical profiles that might be sort of explaining that differences that we're seeing. So essentially not yet, and we're in the process of doing that. But again, like I said, visually they look quite different. So there might be more there that we just haven't had a chance to look at yet. I have a question of, your MAGs seem to indicate organic carbon metabolism, but the MAGs only make up a small fraction of all of the metagenome DNA. Do you see evidence for autotrophic processes in the NOG-MAG metagenome data? The answer is yes. There were some from, especially not from just those bends. So those are just the most complete bends. So we do see some autotrophy as well. One thing that I haven't talked about here is that we do have stable isotope probing incubations. And one of the things that we had was 13C bicarbonate, and that some of that data does show that we are seeing autotrophy at these sites. So I don't think that we're just seeing organic carbon degradation. It's just in these complete MAGs, these happen to be the ones that that this were the genes that they had as their carbon source. This question, I don't know if I can answer, but I'll read it out loud. One of your slides, you showed a limit to abundance of viruses at a certain depth. I'm wondering if you've seen similar variation viruses or metagenomic data possibly indicate horizontal gene transfer events across depth as well. That's something we haven't looked at, and that image wasn't from our work. We actually haven't looked at the viruses in these samples, so I'd be really interested if we collaborate in the future with someone that would be interested in looking at the viruses here. Currently, we're focusing more on the bacterial and archaeal side of it, so I don't really know, at least for our data, sort of where these sort of limits might be. I'm sure they're there. I don't see any reason why we wouldn't have any viruses there. We just haven't gone into that yet. So I actually now need to leave. So I will say thank you for listening and for your questions. I'm sure my email is potentially available if anyone has any more questions. So thank you very much and for tuning in, and I will say goodbye.